Writing's not that easy, but Grammarly can help. This sentence is grammatically correct.
Programs are conducted by professional organizations. Learning goes beyond the classroom with post-dinner sessions that include rigorous exercises. To encourage innovative student projects, seed funds are provided by the Institute and projects are mentored by industry experts. No wonder then that BIT has been consistently ranked as one of Anna University's top-ranking institutions for the highest pass percentages as well as number of ranks. ISTE New Delhi awarded the Bharatiya Vidya Bhavan National Award for an engineering college having best overall performance for the year 2009. BIT also boasts of A-grade accreditation from NAAC, NBA accreditation, ISO 9001-2000 certification, and Good Green Governance Award. Bannari Amman Institute of Technology is backed by the Bannari Amman Group, a premium conglomerate with net worth exceeding $210 million and sales turnover crossing $490 million. The group is a dominant player in all the sectors in which it operates: manufacturing and trading of sugar, granite, textiles, garment, distribution of automobiles, power generation, healthcare, real estate, education, and many others.
It is our utmost passionate endeavor to provide the highest level of technical education in a world-class environment to our students. I personally invite you to come and share the BIT experience.
Situated on the banks of river Bhavani at Satyamangalam in the state of Tamil Nadu, a 176-acre picturesque and serene atmosphere surrounded by the Nilgiri Hills. 16 lakh square feet of spacious buildings, well punctuated with courtyards and pathways. Smartly planned academic blocks with well-furnished lecture rooms and computer centers. State-of-the-art laboratories.
Dear participants, please kindly mute your microphone and video off. Participants, kindly mute your microphone and video off. And stop sharing the screen.
அதே அடுத்தவங்க போட்டாங்கன்னா உங்களுக்கு போயிடும். அதான் பிரச்சனை.
Sashi? Right. இப்ப Lakshman sir. Hello, Lakshman sir. Lakshman sir, stop presenting your screen. Lakshman sir, stop presenting your screen. பாருங்க அவர் பண்ணவே இல்லை. Dear participants, able to hear me? Am I audible? Dear participants, am I audible?
Yes, yes.
Okay, done, sir. Done, sir. Done.
Yes, ma'am.
Dear participants, now we'll start off the FDP second day. And today we have a chief guest, Mr. Shankar Giridhar Vijay Kumar, is working with Zippo R&D Solutions as a business analyst and data scientist on research informatics and laboratory automation. He holds a university degree in industrial biotechnology from the Government College of Technology, Coimbatore, Tamil Nadu. He has taken up multiple roles over the years in various departments like clinical study setup, clinical data management, clinical data analytics and reporting, clinical data visualization, product development. Before moving to UK to work closely on the lab automation and the preclinical front, now he is in Germany as a senior consultant in data science and digitalization. He has got 6+ years of experience in pharmaceutical industry, contributing to the top pharma's transformation to a paperless world by automating the laboratory principles, using market-leading products, securing the data that is generated by ensuring highest quality and generate reports, thereby supporting in the interpretation of safety and efficacy of a drug, eventually leading to the improvement in the quality of life of mankind. May I now welcome our resource person, Mr. Shankar Giridhar, to address the gathering and make the day a fantabulous one. Mr. Shankar Giridhar, now it is your turn to take over.
Hi, good morning, everybody. Thanks, Geeta ma'am, for the introduction. So, good morning, good afternoon, good evening to everybody wherever you are in the world. I'm Shankar. So, a little bit about myself. So, I work with an e-clinical services company as a data scientist and a data analyst at the moment. So what do we do there? So we provide IT solutions to the pharmaceutical industries. So IT solutions in the sense it might be business solutions, it might be process solutions, it might be automating their laboratory processes and so on. So today we are going to see the applications of mathematics in various services what we provide. So I might be going through all the modelings and the equations on an eagle's eye view. Probably not going into the details of every model that we might see. So I will share my screen now. Right, please let me know when something comes up on your screen. So is anybody able to view the PowerPoint that I am sharing?
No, we are not able to see it. Yeah, now it is visible.
Brilliant. Right. So the title for the day that I've chosen is Health in Numbers, the application of mathematical modeling in multidisciplinary domains. Yes. So as the title says, numbers I'm denoting to mathematics, and health is what I am pointing out as the pharmaceutical industry. So the agenda for the day would be to go through the drug development life cycle and what are the applications of maths in drugs. Say there are two main phases. One is the preclinical phase and the clinical phase that a drug development has to go through, and we can see the modeling in both the phases. And what are the tools that help us aid in achieving the efficacy and the safety of a drug before it reaches the market and actually to the patients. So as you know, mathematics has limitless applications, and pharmaceutical industry is not exempted from that. So so the pharmaceutical industry, the main aim of pharmaceutical industry to develop the design and develop drugs for the improvement of the quality of life of mankind. So for a drug to be prepared, an average time span of 12 to 15 years is taken before it reaches the market, before it's been approved and reach the market. So which means the pharma companies are investing close to $2 billion, which is 1.5 lakh crores INR, which is quite a lot of money. And out of 10,000 plus tested compounds, only one might be a candidate for the drug. Only one compound might be a potential drug, and that might reach the market. So only 3 out of 10 drugs pay back the costs for the pharmaceutical industry. So as you can see, there are different stages the compound has to go through before it becomes an actual drug, and it's being approved and launched into the market to be able to be available for the patients. So I will go through each and every stage on a very high level. I'm not going to go into the details. So the different levels that comprises of multiple stages are the discovery stage, the preclinical stage, and the clinical stage. The clinical stage actually comprises of phase 1, 2, 3, and 4. So So this is what I split into two. So one is the preclinical stage, which includes discovery and preclinical, and one is the clinical stage, which includes all the 4 phases of the clinical trials. So the first stage is the target discovery. So what is a target? A target is actually a protein that is associated with the disease, or a microorganism that contains the proteins with the disease. So when you analyze closer in a laboratory, all these yellow dots that we might see are the normal proteins that are inside the body, and one blue dot that you're seeing is actually a diseased protein. So which we call the target, to which a drug needs to be discovered. So all these processes happen inside the laboratory, and multiple experiments are carried out in order to search for the target. So the proteins are able to talk to each other through cellular networks. And they are able to transmit their messages, their impure messages, their pure messages to the adjacent proteins. So this way, transmitting the disease. So the base point of the starting point of the transmission is identified as the target, and the hit compound and what we call the potential drug candidate is being identified for that particular target. So this is the target. The next step is the hit. So as you can see, once we've identified the target, the next thing is to identify a potential drug that binds into the target and alters the diseased protein and gets it back to the normal state. So a compound capable of binding to the identified target is discovered, and the compound will be evaluated if it modulates the target in a way that alters the disease. So the alters the disease means it might bring back the protein to the original state, or get rid of the diseased disease that's stuck onto the protein. So once that's been evaluated, you get to evaluate the safety and the effectiveness of the compound. So it might be a cluster of compounds that goes and binds into the proteins. It might be one single compound, and after you get the actual compound that is binding to the protein, you might enhance with binders and others for effective binding and evaluate it for safety and efficacy. So the next step is now we have a target, now we have a hit, the compound, the potential drug, and now we need to select the actual candidate. So out of all the compounds that bind to the protein, say I was telling you, right? So 10,000 plus compounds might be binding to the target, and only one might be a potential drug. So we are conducting further experiments to predict the ADMET properties. So ADMET is actually a very common term that's used in pharmacokinetics or the drug discovery methods. The ADME is the absorption, distribution, metabolism, excretion, and toxicity. So it is actually the rate of absorption, distribution, and the processing and the excretion of the drug inside and from the body. So this is how we calculate or evaluate the safety of the compound, the identified compound. And once it passes the ADMET properties, or it has appropriate pharmacokinetics, that is considered to be safe to be administered into the patients. So before it's being administered into the patients, the regulatory authorities like the FDA, the Food and Drug Administration of the USA, it's an example. There are multiple health authorities.
Mr. Giri, just a minute. Mr. Giri, just a minute. Sir, yes. Lakshman sir, please stop sharing your screen. Lakshman sir, please stop sharing your screen. Professor Lakshmanan, please stop sharing your screen. Professor Lakshman, please stop sharing your screen.
I should be able to override his presentation, ma'am. Let me see.
Geeta, right, madam.
So my screen.
Yes, ma'am. Tell me, ma'am.
You can remove, the host can remove that participant.
Yeah. It has been done. It has been done.
Yes, ma'am.
Okay, thank you.
Yeah, so my screen is back once again, right?
Yeah, you can go ahead.
Yes. Yeah, so to where was I? Right. So before administering the drug candidate or the potential drug into the patients, the health authorities might want to test more, for which we use the animals. So the drug testing inside the animals is being done inside the labs. Multiple experiments are being carried out. Yes. So the new drug needs to be tested in animals before humans. So why do we test in animals? One is the health authorities require the testing of the potential drug in animals, whereas the other reason is, so if it is a simple disease, if there is a small modification in the protein, it might be identified in experiments, in the laboratory experiments itself. But if it is a complex mechanism, if it is a complex disease mechanism, it can be understood only by animal studies. And after thorough examination and testing inside the animals, we do the human clinical trials. So when I talk about animal clinical trials, the preclinical trials, so to say, it is done under extreme regulations. The welfare of the animals are also taken care of. Right. So the next step would be to actually administer the drug into patients. Into the patients. So the clinical trials consists of 4 phases. So one is, it's pretty normal. So it's phase 1, 2, 3, and 4. So phase 1, the drug candidate or the potential drug is administered to healthy volunteers to determine the safety. In phase 2, the sample size, so to say, is increased. So it might be 100. Yeah, just a moment. Stop presenting. I'm going to share again. Presenting window. Are you able to share my screen and see my screen?
Yeah.
Yes. Yes, where was I? So the phase 2, the sample size, so to say, is increased in mathematical terms, so the statistical terms is increased. This will evaluate, this will further prove the drug or help us in evaluating the efficacy, which we say the effectiveness, and the dosage of the drug that needs to be administered into the patient for the disease. So while doing phase 2, the adverse events, so to say, or the side effects are also evaluated.
Ma'am, please share the screen, PPT screen.
Are you not able to see my screen?
No, sir.
I'll share again. Just a moment.
Madam, pin to the speaker so that you can view the screen. You please pin to the speaker, madam, to view the screen.
Yes, I got, ma'am. I got. Thank you.
Brilliant. Right. So in phase 3, the investigational drug, the new drug. So the potential drug is now the investigational new drug, and it's now administered to a bigger group. So so it might be administered to a bigger sample size of 1,000 to 3,000 patients, and the right dosage, which was determined in the previous phase, is also administered. And now we assess the safety and efficacy, monitor the side effects, and compare with other treatments that are already available in the market. So it might be a different drug from a different pharmaceutical industry for the same disease for which a drug is already available. So the next step is to document all the all the findings that we have from phase 1, 2, and 3 and put it as a proper document and submit it to the health authorities and get it approved from them, and the product is launched into the market globally. So as you can see, the sample size, which was started from 10 to 15 healthy individuals in phase 1, has now grown into thousands and thousands of people in phase 3, and now it's been released globally. So the sample size will be a representation of the global population. So it's not going to be a population from a single region. So it might be patients at multiple sites at multiple places across the globe. Right. So after releasing or launching the drug into the market, post-authorization safety updates are being updated to the health authorities on a regular basis. Any additional information that they also require are also updated on a regular basis. So this is to this is to actually evaluate the safety at a long term and for a bigger participant group or sample size. So this actually covers an eagle's eye view of the drug development and design process. So where is mathematics and mathematical modeling or statistical modeling involved at any stage of drug development? Pretty much everywhere, I would say. So mathematical modeling starts from target discovery until the approval or the market phase. So I'm going to take a very important aspect of the drug design, which is called the pharmacokinetics. So as we saw earlier, pharmacokinetics is actually involving the metabolism of the drug or the drug candidate or the potential drug that is administered into the body. So what is pharmacokinetics, and how does pharmacokinetics help in drug development and design, and what are the models that are used in pharmacokinetics? So the study of the effect that the body has on the administered drug is called pharmacokinetics. So how a body reacts to the administered drug. So what are the basic questions that strike us right away? So how does a drug get into the body, and where does it go? What does the body do to the drug, and how does the body get rid of the drug? So if you remember, the 4 basic elements of pharmacokinetics are A, D, M, and E: absorption, distribution, metabolism, and excretion. So let's start by plotting a simple graph. So this might look simple, but it is very, very powerful in pharmacokinetics. So so we have a concentration of the drug on y-axis and the time in x-axis. So if you consider 0 as the state in which the patient is being administered, so once the drug is administered into the patient, there is a curve. There is the travel or the trajectory of the drug into the body or inside the body. So starts from 0, goes to a saturation point, and starts falling from there. So it's similar to a bell curve, I can say. So there are multiple terminologies that can be derived, or multiple equations that can be derived from this curve that help us identify a lot of parameters in the drug design techniques. So so the first phase, where you can see a double-headed arrow down, more drug is taken in than it is excreted out. In the next phase, more drug is excreted out than it is retained inside the body. So the saturation point, where it starts to fall, as well as where the drug starts to show the therapeutic activity, the point is called the concentration maximum, which we call the Cmax, and the time taken to attain Cmax is called Tmax. So this point, the topmost point, is the point where the drug starts to show its therapeutic activity and tries to combine with the target and tries to alter the target, the disease. Next, from the point of Cmax, at the point where 50% of concentration is lost. It's called half Cmax, and the time taken to reach half Cmax is called T-half. So basically, T-half is the distance between Tmax and T-half. It's not the distance between 0 and T-half. It's the distance between T-half and Tmax. And you can see the area that is actually under the curve is the total exposure of the body to the drug, so which we call the bioavailability or the F factor, so which you will see in the future slides. So this is bioavailability means the availability of the drug or the retention of the drug inside the body for which to carry out the therapeutic activity. Right. So this is a very, very simple graph, yet a very powerful graph. Why? I will tell you in the future. So some of the applications of clinical pharmacokinetics. So that graph that you saw before in the previous slide is actually used for so many applications. So one is design and development of drugs with lesser side effects, optimum formulations for better use of drug, targeted and controlled release formulation, multiple dose regimen. So multiple dose regimen is actually the dose frequency and the dosage of the drug. So the next is the selection of appropriate concentration of the drug, appropriate dosage of the drug, appropriate route of administration, the right drug for the right illness, and to predict drug-to-drug interactions when there is another drug that you have already taken. So what is the interaction of the new drug that you are taking and the old drug that is already existing inside your body? Right. So there are multiple mathematical and statistical models and simulations that are carried out for each of these applications, starting from 1A to 3. There are individual mathematical models. So let's take one of these applications and see them in detail. So I am taking the multiple dose regimen as an example and show you the mathematical models and the equations that are available to determine the dosage of a drug. So when we are talking about dose regimes of a drug, there are two terms which are quite important. So one is the steady state concentration and one is the dose interval. So steady state concentration is the concentration at which the drug is actually showing the therapeutic activity. So if you remember the previous hand-drawn chart or the graph, so the Cmax is actually the point where we achieve the therapeutic activity. So in order to achieve Cmax or in order to achieve the steady state concentration, there needs to be multiple administrations of the same drug until it reaches a steady state and show therapeutic activity. So if you see the first graph, the red lines, the red curves are the ones where it is exponentially increasing to reach the green curve, which is the steady state concentration, and where the x-axis shows you the half-lives. So at each half-life of the drug, you administer the same drug again. So you will reach another Cmax, and you'll reach another T-half, and so on, and you'll reach a point where the Cmax is going to be constant, and it's stationary here at the green curve. So this is where the drug will show therapeutic activity. And in this graph, the dose interval is calculated on the basis of half-life of the drug. Increase or decrease of the dosing interval make changes to the average drug concentration attained in the body. So in this graph, you can see the therapeutic window. So right from the first administration until the last administration, this is what we call the therapeutic window. So these two terms are quite important in order to determine the dose regimes of a drug. So model-based design approach of dose regimen, IV bolus dosing. So IV bolus dosing is intravenous bolus dosing. Intravenous bolus dosing, you may call it a simple injection into your intravenous system, or it might also be an oral injection, oral administration of the drug. All right. So there are basically 4 steps in order to derive the dosage of a drug. So one is calculation of the steady state concentration. So the steady state concentration will be equivalent to is calculated by the upper concentration and the lower concentration. So the upper lower concentration is the Cmax of the first administration, and the Cmax of the last administration is C lower and C upper respectively. An estimation of the dosing rate or the dosing interval is actually a product of clearance into steady state concentration divided by bioavailability. So the bioavailability of intravenous dosing will be considered as 1. So which essentially makes this equation clearance into CSS, the steady state concentration. So what is clearance? So the clearance is actually the rate of clearing the drug that is being administered into the body. So it might be calculated in mgs per liter, so hours per liter. So you will see a practical example in the next slide as well. So, and the next step is to calculate the maximum allowable interval. So this equation is actually calculated based on the half-life. So it's based on CSS max, CSS minimum, and the K. K is the elimination rate constant. So elimination rate constant is actually a constant which replaces T half. T half is the half-life. Right. So this is the maximum interval between the administration of the same drug to a patient, but it is typically the administration occurs before attaining the maximum allowable dosing rate. The next step is the estimation of dosing. So the dosing rate into the dosing interval will give you the actual dose that needs to be administered in real patients. So all these steps that you see are being derived through computer-based modeling or statistics-based modeling. So there are multiple tools that have these equations and models embedded inside them in the IT industry, which data scientists like me might use it and pull in the right model from the right repository in the tool and use the right model that is being defined in the protocol. So we actually match what is defined in the protocol and what is available in the repository and calculate the dosing. So similarly, we calculate other applications as evaluate the other applications of pharmacokinetics as well. So what we are seeing now is actually, say, less than 1% of what the pharmaceutical industry of what the mathematical applications of are available in pharmaceutical industry. So so as I said, there are multiple limitless applications. So this is the practical scenario. So the previous slide has actually had all the equations, and now let's calculate the dose. Oops, I'm sorry. Let's calculate the dose of a drug called theophylline. Theophylline is actually a drug which is used in chronic respiratory illness. So there are some numbers that are already given, that are already being determined. So one is the therapeutic range, which I mentioned it as the therapeutic window, if you remember, and the clearance of the drug from the blood plasma is which is 2.6 liters per hour, and the volume of distribution, the volume that is being distributed into the blood plasma, the elimination rate constant, which and the T half. Right. So when you substitute these numbers above in the equations below in all the 4 steps, you will derive at a dose which is equivalent to 300 mg. As you can see, the last but one line, which shows you 300 mg of the dose needs to be given. It's the appropriate dosage of the drug theophylline. So the next line states something called as the loading dose. The loading dose is actually a dose of the same drug given at a higher dosage in order to reach the half-life rapidly. So if you remember clearly from the previous slides, the longer the half-life, the longer the time taken to reach Cmax and exhibit therapeutic activity. So the loading dose will actually make the drug to, or the potential drug to attain the Cmax quickly and show therapeutic activity, so that the drug can stay inside the body for a lesser time and excrete it faster than usual. Right. So this is a practical scenario. So there are multiple tools. There are multiple tools that do these equations, and for each route of administration. So what we saw was the intravenous bolus. So the first one was the intravenous bolus administration, and there are multiple routes of administration of the drug. So it might be continuous intravenous, it might be extravascular, it might be oral, anything. For each routes of administration, the dosage interval, the loading dose, and other equations are separate or rather different. Right. So as I said, there are multiple tools that aid in these mathematical modelings, and one such main tool is the electronic laboratory notebooks. So all the experiments that you that happen in the preclinical phase are typically in laboratories. So people have instruments, people have softwares, people have papers and pens. They have the samples, the microscopes. They do all sorts of experiments, note down the details, the data from the instruments, and load the data into the tool, and the tool will compute statistics for them and give out the, let's say, pharmacokinetics in this case. So the electronic lab notebook is one such industry-leading tool that does maximum statistics and statistical modeling, that includes or has embedded and GraphPack, Prism, ImageJ, ClusterLex, and Microsoft Excel has add-ons that support curve fitting of pharmaceutical data. So where you plot the data of a particular potential drug candidate in order to evaluate safety and efficacy of the drug. So this pretty much covers the first phase, which is the preclinical phase, where we've seen about the pharmacokinetics of drug administration, and what are the different different statistical or mathematical computational models that are involved in one of the applications of pharmacokinetics. Right. The next big stage or the big phase is the clinical data, the clinical trials. So now that we've covered a preclinical phase, we are now going into the clinical phase. So now that the preclinical testing and the testing inside animals is complete, the next step is to administer the potential drug inside humans in clinical trials. So what happens is you collect a lot of data from the hospitals or from any sites, and standardize them, group them logically into tables or data sets, and analyze them for safety and efficacy. So again here, safety and efficacy plays a main role in order for a drug to reach the market and to provide and to be proficient in order to handle a disease. Right. Then the next step is to report the safety and efficacy analysis. So we usually do tables and figures which consist of descriptive statistics or categorical summary. So the categorical summary is giving the count of all the subjects and the percentage, and the continuous summary will give you the count, mean, median, ST, min, max, etc. The tables and figures are created based on the statistical analysis plan drafted by the biostatistician. So one important person, one important role in the clinical phase is the biostatistician. So biostatistician play a key role in drafting a statistical analysis plan and deciding what statistical test needs to be used in order to measure or evaluate the safety and efficacy of the drug. So the biostatistician decides which statistical test to be used based on a number of factors, like data that is being generated out of the sites, the sample size that's being tested, and the purpose of the drug, obviously. So some of the examples include chi-square test, Fisher's exact test, and regression analysis, etc. So all these tests are once again embedded into the IT tools that we have, and you just feed in the data and you get back the charts and figures with all sorts of analysis, and you can interpret results out of it. Right. So some of the examples that I wanted to show is a descriptive statistics table. This is how a demographic and baseline characteristics table would look like. So you would, in order to perform clinical trials, you would actually get the demographic details of a patient, like what is the age of the patient, what is the sex of the patient, what is the race. So all these factors determine the functioning or the operation of a drug inside the body. So a body is different region wise. So I might not have the same body as an American has, by metabolism, and they might not have the same body as an Australian has. So, and region wise and also race wise, if it is an Asian, if it is an African Asian, if it is Hispanic, and all this races are also collected. So they might have better. So some people might have better resistance, better immunity, or they might be less immune. All these, they might have more side effects, more adverse events, so to say. And what is the sex of a patient? So what happens to a male? What happens to a female? What happens to a pregnant female? So how is it affected, and what is the age range? So what happens to patients below 30 years and between 30 and 50 years, and what happens to the patients after 50 years, and so on. So all these factors determine the operation and the metabolism in the drug discovery. So this is one descriptive statistics table, and another example would be to have the descriptive statistics and the p-value. So the p-value, as you know, is the probability. And the probability has an arbitrary, so there is an arbitrary number 0.05 below which the drug is proved to be effective, and above which a drug is proved to be less effective. So that's an arbitrary number, and there are a lot of statistical models that prove that 0.05 is okay enough to evaluate the safety and the efficacy of the drug. So these are the factors that factors or characteristics of the study population that determine the operation and help us aid in evaluating the efficacy and the safety of a drug candidate. So similarly, like tables, we would also generate plots like the bar charts, the scatter plots, the pie charts, and any other graphs. So this we basically call it as the data visualization, and the visual data is more effective than written down data or computer data, so to say. So you will be able to see the outliers, or you will be able to see the clusters. So the groups of clusters, and so basically you can plot categorical data into bars, and you can plot quantitative data as say scatters. And you might also have box charts, pie charts, and whatnot. So you might have all these for the analysis and the evaluation of safety and efficacy of the drug. So what are the applications of the reports that we just generated? So clinical data, we just generated tables, graphs, figures, and the applications are the report data are compiled as a part of the statistical analysis plan and passed on to the group of biostatisticians again in order to make inferences or interpretations, so and predictions. So reports are analyzed against the statistical tests that were proposed, and interpretations are submitted to the health authorities for approval. So let's say there is a drug that's proven to be effective against cold or cough or fever. So you carry out a series of statistical tests in order to prove that the drug is effective and safe across all regions, for all races, and for all genders. And it's being submitted to the health authorities, and they approve it. And only after that, you get the drug to be launched into the market globally. So after which you give post-marketing, post-launch numbers. So post-launch numbers are for the safety analysis on a longer term. Right. Some of the tools that help us aid in data modeling and visualization. Some of the tools might already be known for you: Spotfire, SAS, R, Python, Oracle, Tableau. So Oracle is quite special here because Oracle is a database, and all the data that's being generated across all the sites globally are being stored in databases. So Oracle is one such database where all the data might be stored, and you pull the data from the databases and write scripts and give stats, and you put in statistical models to get out the figures and tables. Right, that's it. We've covered the design and development of drug design and preclinical phases, and we've seen about pharmacokinetics and what are the models that are used for determining the drug dosage, and we've also seen a gist of what happens in the clinical trials. Thank you very much for your time and patience, for listening to me. You've been an overwhelming audience for me. Thank you so much. Thank you so much for the opportunity, Geeta ma'am. It's been a pleasure to present.
Most welcome, Giri. Just a minute. Dear participants, any queries, if you would like to get it from the resource person, you can get it. A query session now.
Hello, sir.
Hello, ma'am. Yeah.
Sir, I'm Usha. Sir, how the impulsive differential equations applied in pharmacokinetics?
Right. As I said earlier, so any differential, any equation of that sort, a statistician or a biostatistician might be able to answer you precisely. So this question, I'm writing it down, I'm noting it down and sending it to the biostatistician. So how impulsive differential equations are being applied in pharmacokinetics and what helps in the drug design, and sending it to them, and I will get back to Geeta ma'am with a response, and she might let you know the response.
Fine, sure. I'll let the participants know about it.
Yes. So we are working as data scientists. So we usually work with large data sets and compute statistics, or the statistical models are available in the tools that we use, like say R or Python or Spotfire or any such tools, and we just use the data, the model that's being preferred by the statistician or the biostatistician, and get the data out and the statistical data out for them to analyze more.
Still, any more queries if you have, you can get clarified. If not, we can just conclude the session. Dear participants, any more interactions you need? Okay, fine. I'll take it as nothing is needed more than this. So now I thank our resource person, Mr. Shankar Giridhar Vijay Kumar, for giving such a wonderful presentation. He has rightly pointed out the application of mathematical modeling in biotechnology field. He has given a very good presentation of his work through normal curves, and also explained how the mathematical model can be used to derive the dosage of the drug. He has also elaborated how to use chi-square test, F-test, regression analysis through softwares in order to get the desired results of the patients. We could closely see how the mathematical tools have been used in biotechnology field. Finally, to conclude, he has given a fantastic presentation. Thank you, Mr. Shankar.
Thank you very much, Geeta ma'am. So I'll share this presentation with you, and you can share it with the participants if needed.
Yeah, sure.
Brilliant.
And thank you for sparing your valuable time with us and giving all the inputs, how these mathematical tools have been used into your field. And still, if any queries, I'll let you know through mail, and you can just clarify it for the participants.
Yes, thank you very much. So thanks everybody for being a brilliant audience. I hope everybody are staying safe and healthy. And see you. Have a great day.
Thank you.
Good morning, ma'am. Will you send the feedback form and chat box?
Sir, already it has been sent. You just look into the chat box.
Okay, thank you.
Feedback link is given there in the chat. Yeah, sir.
Madam, can you send it again, please?
It has been sent. You just see, ma'am.
Ma'am.
Yeah, ma'am. It has been sent.
Yes, madam. Okay, thank you.
Yes.
Ma'am, Geeta ma'am, can I get yesterday's feedback, ma'am? I could not able to fill it.
Ma'am, yesterday's feedback we don't have right now.
Then how to submit it, ma'am? Because yesterday after the session I had to leave for some work. I could not able to fill it.
Yes, ma'am.
Okay,
hello, Girija sir. I am Ponal Mata.
Hello, ma'am.
Ma'am.
Tell me, ma'am.
That's what feedback from yesterday said.
Ma'am, I have one question, ma'am.
Yeah, the resource person has left, I think so. Just a minute, I'll just see whether he's there in the group or not. No, he has left. He has left.
Or will it be possible to send it to my mail ID, ma'am?
Hello, ma'am.